So, good afternoon, everyone. If you are in the building or on site, please come to the auditorium. It's much more fun in person. Otherwise, enjoy your food and coffee sitting at your desk watching this. So we're in for a good one today. We have Matthew Gilmour, who's the group leader of Microbes and Food Safety Research Programme at the Quadram Institute. Matthew's got quite an impressive background. A Canadian whose previous role was the Scientific Director General of Canada's National Microbiology Lab. And through that, he's co-chair of national and international public health networks and implemented genomic technologies in health systems. It's an enviable CV already. He's a board certified, sounds strange, board certified, you're a certified clinical microbiologist, should be certified. And the hospital's laboratory lead for infection prevention and control. PhD training was at the University of Alberta, where we looked at genomics of cell biology of conjunctive bacteria plasmids that carried antibiotic research traits. He's a multiple prize winner. I won't go over that. You'll have seen that in the text already. But today we're talking about food, a key point for everyone. And Matt's going to talk about the genomics for food business, a case study of persisting Listeria. Matt, the floor is yours.
Derek, thank you very much for the kind introduction. Raman, thank you for the introduction, for the invite to come up. It's been a remarkably informative day. So Raman and I have toured the new facility. Congrats to everyone on the new institute, the new real property that you have here. It's fantastic labs. It's lovely to see all the other buildings that you have. So everything related to the barley. We just got back from the crop systems, the vertical farm. It's a really impressive campus. Actually, I think I've learned more about your campus this day than I have about the Norwich Research Park, which I'm joining from, where I've now been for 4 years. On the Norwich Research Park, I'm a group leader at the Quadram Institute. We ourselves are a brand new building. We stemmed from the old Institute for Food Research, but Quadram's been given the mandate as a BBSRC-funded institute to help improve the quality and the safety of UK food supplies. So there's kind of a yin and a yang to what we do at Quadram. On the health side, it's about putting nutrients and vitamins and improving the nutritional quality of UK foods, and then moving over to a gut health component. So with that improved diet, how does that impact overall gut health? And for our side, that I'm joining from, the food safety side, what we're looking for is, instead of improving health, we're trying to take risks out of the food supply. So we're looking at predominantly microbial risks. So the organisms like Salmonella, Campylobacter, or Listeria that continue to find their way into food systems, whether it's from an agricultural perspective, whether it's spillover during food production, but nonetheless, these are organisms that are still finding their way into the food chain and then presenting as a human health risk. I will do a case study on Listeria and how we're using genomics in tandem with food businesses, and very science-focused food businesses that want to, again, remove and get a handle of these threats, because, again, they are likewise frustrated that they continue to find their way into their products. But I'm going to start with a perspective that actually connects to a lot of things that I heard here today. Being a BBSRC funded institute, one thing that they asked us to do was to form, a couple of years ago, the Food Safety Research Network. This is meant, just in its simplest form, is just to connect food businesses together with researchers and potentially government over questions, whether it's proof of concept, whether it's other research questions that they have related to, again, microbial risks. So this isn't about allergens, it's not about chemical contaminants, it's again about the Salmonellas, the Campylobacters, the Listerias, the toxigenic E. coli's that keep finding their way into the food system, but the producers want to get a hold of it. Of course, food businesses aren't often eligible for UKRI resources. Sometimes they can get it through Innovate UK, which is wonderful. In terms of BBSRC, it's a tighter window for them to get in. And this Food Safety Research Network is just a clever way to actually give them access to those types of resources, but more so give them access to the partnerships they need to answer the science questions that they have. In terms of why food businesses have been interacting with us, is because, and we love this line from FAO, WHO, is that if it's not safe, it's not food. So that connects through to all the interest that we all have in sustainability, that food can be as nutritious as you want, but if it's contaminated with an allergen, if it's contaminated with a microbe, then it's not fit to be served and has to get wasted anyways. So yes, food safety is important for piece number one. We want to keep the illnesses away. but it's also that cornerstone of a healthy diet. Of course, for food producers, again, we've got the sustainability piece, it reduces waste, but by producing safe food, it's a reputational piece for them and they gain a lot of access to new markets. So yes, there are some food businesses that I will admit want to spend as little as possible on their microbiology budget, but there's there's many more that see this valuation towards why it's important to produce safe food. We've been working a lot with Innovate UK, and they just produced this fantastic white paper or strategic piece on food industry priorities for a sustainable food system. And within this, food safety lands on this map as well too. And this is something that's common that we've seen, and one of the reasons why BPSRC asked us to form the network was food safety might not always be the kind of the headline piece in a lot of components of agri-food, but it's always part of the conversation. And that's what they've asked us to do is to feed businesses opportunity to pursue that culture of food safety. This is a great position paper from the Global Food Safety Initiative that just talks about the different components of why a food business is going to have it as part of their culture. They're going to want to train their people on food safety. They're going to want to have engineering controls in their building in terms of food safety. We just saw that at IGS across the way. It's half of what they're making. They may not use the word food safety, they use the word hygiene, but it's the same thing. They're trying to produce safe, clean, hygienic crops there, but it's a lot of engineering that goes into it. It's a lot of innovation and research. So it's great to see a business like that's putting this forward. In terms of how we've gotten a little bit of traction as the network, this is actually feedback coming from our members, is that we listen to them, we ask them what they want to prioritize, what they have as research questions, we help them prioritize, but importantly, we connect them to the actual teams that can help them accomplish their research questions. Because as a food business, you may have technical managers, but you're not going to have microbiologists, you're not going to have data scientists potentially to help you get there. But we get people connected to that. Once the work gets done, we help them report some impact and promote success. One thing that's not on this is the resource piece. Again, we're resourcing projects that people might otherwise struggle to get pounds around. But that first fundamental piece in terms of the listening is just asking a very simple question of what are the problems we're solving by us as a community, as a network of food businesses and researchers. Where is the money best spent? I'll go over this very quickly, but it's ultimately hearing from food businesses. It was 4 priority areas that they were concerned with in terms of microbial. The first one is the obvious. It's the known pathogens. It's again the salmonellas, campylobacters, and listerias that continue to find their way into the food chain. But how can we actually think differently about acting against them? Priority area number 2 is the novel and emerging ways of producing food, whether it's alternative proteins, or whether it's novel plant-based products, whether it's vertical farming. This is areas of perceived risk. There potentially isn't risk there. There's actually opportunity to control those risks, but there's perceived risk in these new commodities or these new technologies. The business wanted to prove that what they have was safe. The third piece was applying knowledge, so making sure everything from environmental health officers to their own teams have the information that they need. And the fourth piece is safety. So how do you actually empower consumers? You've made safe food. How do you empower consumers to keep food safe within their own house setting? So I'll run through some very quick case studies on those 4 priority areas. Then I'm going to get through to the Listeria piece. This all connects through because it's all coming from the voice of food businesses. The most common thing that we heard from food business was interest in the application of bacteriophage. So this is Greek for bacteria eater. They know that this is an alternative antimicrobial that's coming out that can be used to control these microbes within different settings. Working with the Phage Innovation Network at Innovate UK, they put together this lovely piece in terms of how can we use phage. It's not unilaterally towards food safety. It can be for animal health, human health, so precision medicine, but also, of course, agri-food settings as well to reduce pests. One of the main problems with, one of the main challenges within phage in the UK, actually internationally, people would say this, that there's a lot of science in the UK related to phage, yet we're kind of below the curve in terms of implementation and adaptation. And the reason for this, and this just came forth in another white paper that came out of the group, was that there's a lot of barriers in terms of regulation. Again, animal health, human health, food, environment, these all have different regulators. So how do you push, if you're a small business, how do you push through those different regulators? The actual bio process, the ability to produce phage en masse in the UK is a little bit low. So we've got a capability issue, we've got a regulation issue. This is all detailed in this, but it gives us a roadmap to start to get phages into common use. This is very hot off the presses. So last week within London, we hosted a one-day workshop on the diagnostics of Shiga toxin-producing E. coli. So Nicola Holden, who I believe is an alumni of James Hutton. So with Nicola Holden, the Chilled Foods Association, UKHSA, Food Standards Scotland was a co-organizer of this. What the impetus for this was the large E. coli outbreak that happened presumptively in leafy greens earlier this summer. So there was 2 deaths, 100 hospitalizations, 300 illnesses across the UK. But even with that scope of illness, it was never actually confirmed what the actual source was. It was presumed to be leafy greens that were in salads and wraps. But that presumption is based upon the technical challenge that it is to actually detect and confirm toxigenic E. coli in the lifespan of a short shelf life product like a leafy green. So this is where the food businesses, industry, governments, and the testing technologies all came together to actually identify this as an urgent need, discuss what would be the ways to advance through the challenges that we have right now to not have that we don't have actionable testing for Shiga toxin producing E. coli. So we had the Fresh Produce Consortium on the stage, we had the Chilled Foods Association on the stage, FSA, UKHSA, and then a variety of the actual contract testing labs that can actually provide diagnostics for toxigenic E. coli, each of them readily admitting that it is a challenge, again, to do this in a cost-effective, timely manner. So again, very hot off the presses. I haven't even had a chance to clean this up because it happened last week, but we came up with 4 potential work streams that would help improve this. Again, I'm showing this, again, this has nothing to do with Listeria, but this has to do with the concepts that we've been talking about all day long, but how institutes like ourselves can work together and advance through what are some really challenging pieces. And the first piece that we talked about on toxigenic E. coli was the sampling scheme. When do you actually sample in the field on a crop? When do you sample the water supplies? How many SKUs do you sample once it's been harvested and packaged? But you need to have some sort of sampling scheme to identify that you do have good agricultural practice, confirm that you do have good manufacturing practice, that there's an absence of risk going to the market. Because if we're just waiting for people to get sick and then working backwards, that's not the best process. So instead, let's have a sampling scheme. Let's have one that's based upon not just a routine piece, but also climatic triggers. So what are the rainfall events that would dictate when there's more sampling needed? What are the economic events, like changing in consumer profiles that would dictate when you want to have more sampling? This all needs to get factored in. I mean, it's not something that I can do alone as a microbiologist, but as we start to string together the expertise of institutes like ourselves, this is going to work. Then we get off to the second piece, which would be the testing methodology. Right now in the UK, it's a bit challenging because to actually confirm a Shiga toxin producing E. coli, you need a Cat 3 lab, so some elevated containment. So a lot of the testing for this occurs offshore. It's technically challenging to do because you got to sift through all the other E. coli. Anyways, there's opportunity to improve the testing. Derek, we're talking about this in your office, the lexicon, that shared understanding of data. In terms of actionable findings between a food business and a government authority, you get kind of things like presumptive findings, confirmed, DNA detected, and it really changes how people are able to actually action some of this information. So have a shared lexicon and start sharing this data amongst each other. So you start having a baseline of what is the incidence of this organism, whether it's, again, in farm settings, whether it's in food production settings, whether it's in food itself, or whether it's related to human illness. Get that baseline so we can start to figure out how this organism is transmitting and entering the food chain. So that you've got that framework for interpretation and actions.
Louder, right? Have trouble. Microphone. Is it coming straight? You can try that. If not, we can.
Sure. Okay, so moving on to the second piece, and we just saw this 10 minutes ago. So the vertical farming again. So we've got priority area number 2, which is the novel and emerging technologies. It's fantastic because you've got controlled environment agriculture. You've got a chance to have everything under control in terms of your water, in terms of your substrate, in terms of your light, in terms of the absence of, we were talking about, there's no birds flying over, pooping on your crops, there's no foxes running through. You've got every chance in the world to control the environment, but do you have the engineering pieces in place? So this is working with the Fresh Produce Consortium, it's working with Farah Science to actually come up with best practices in terms of, so this is business to business best practice on how to actually establish and run a vertical farming operation with those hygiene controls in place. What are the engineering pieces? This is where a group like IGS can easily contribute that. It's a pre-competitive piece. They don't have to contribute anything related to IP, but there's a lot of vertical farmers who want to make sure that their entire industry is set up for success. They don't want any of their members falling down, because the last thing you need is the Morrisons or the Waitroses of the world. Again, there's risk within this food category. So they all want to build things together, and this is work that we're funding through APGC and FARA. The last thing I'll mention before we get on to the Listeria is a piece that we're building right now. This is not live, but a lot of food businesses came together and said, wouldn't it be wonderful that if, as a business, that we could share our own microbiology data with other businesses? Of course, there's regulatory data, there's data that they have to send up to regulators, but they never see what's happening in other businesses unless there's a problem somewhere that's marketed. What they want to do is have a system where, if we're all testing different components of our supply chain, if we can put that into one place, then we'll understand where risk is present in the supply chain, but also where risk is absent in the supply chain. Because it's going to be very uneconomical if we're all testing, I'm pointing at people in the room here, if we're all food businesses and we're all testing our supply chains independently, that's not economical, and we're probably not going to get on top of risks if something does enter the system. So this is where businesses do want to come together. We've branded this, again, notionally, the Food Microbiology Intelligence Network. If anyone's familiar with food authenticity, there's a food industry intelligence network where they're already sharing authenticity data. And so, yes, we're trying to create a trusted network from a business-to-business perspective to share and co-analyze data on microbial risks and to get those sector-wide insights that would allow collective actions, early actions to safeguard consumers. Once you actually got this going, you can not only look at the present data, but you can get that baseline piece, the historical. So where have things been found in the past? Where are they entering the system now? You do this long enough, you can actually start to get into horizon scanning, where you can start to get a little bit predictive. Derek, you used this word before, predictive in terms of where things might enter in the future. Again, we're still sorting this out. We've got multiple business signed up, but what we're going to use is Agrino as a system. So this is going to be an AI-based system that will allow for data harmonization, data entry, a data pull from other sources. But it's in development right now, and we've got calls out to businesses to join us into what will be a pilot phase. But you can see this will be a very contemporary way for businesses to actually work together and actually get a little bit of uplift in terms of their own approach to data management. Even if they don't ever learn anything about what's happening in another business, they're going to learn a lot about how to manage their own data for a system like this. I'll skip this one for time. Just a quick note, and potentially online, there's multiple of your own colleagues at James Hutton, Scotland Rural College. We've come together to form a UKRI-funded transdisciplinary network to tackle antimicrobial resistance. There's actually 8 of these networks that have been funded. Our particular one is on agri-food systems, and the whole point here is to pull together multiple different disciplines to reconsider how to study and act upon antimicrobial resistance. So ours is the agri-food network. There's other networks that are related to genomics. There's networks related to climate. There's networks related to One Health. There's networks related to diagnostics. But it's all meant to come together with that collective expertise, again, to really start to manage antimicrobial resistance. I just wanted to mention again that this is partners here at Safari, partners here at Hutton, and it's actually going to officially launch next week. Okay, so we'll get through. What I actually came to do was to talk about Listeria. It was just a decision point. Let's go through some of these other case studies first, because I thought there might be more interest in those other activities. But the Listeria, again, is a learning point on how to contemporarily apply genomics and metagenomics from a perspective that's useful to food businesses. Listeria is a rugged, versatile gram-positive rod. I've been in the UK for 4 years right now, and this is one of my favorite moments. When we first moved here, this was during COVID. We asked the hire company, we said, we're going to go camping this weekend. Can you send something over that would be good for camping and tow a small trailer? And this is what the hire company dropped off. Didn't even have seats in the back for the kids. Anyway, so I'm staring at it in the drive, and I'm like, that actually looks like a Listeria, and it acts like a Listeria. Because that's what Listeria can do, is it can just survive in almost any environment, can go through anything, go over top of everything. And that's why it presents itself as a food safety risk, is that it's very pervasive in the environment. It can get entrenched into food production environments, and then spills over to food, and then presents as a human health threat. We actually just did host the 21st International Symposium on the Problems of Listeria and Listeriosis at Norwich. This is it's one of the rare meetings where the entire community comes together. It's about every 3 years, but both the food safety experts and all the cell biology experts that study Listeria pathobiology. So it's a community of about 200 people, and we came together for 4 good days of science last September. And I'm going to show you a couple of slides from one of the keynote talks. But here is that community, and it's my team in front wearing the green sashes. It was lovely to have and convince people to come to Norwich. It was 200 delegates from 25 countries and 5 continents. The Listeria itself is actually, in terms of its age, it's a relatively more recently recognized foodborne illness. It wasn't until the mid-'80s, when there was a lot of incidents related to contaminated cheese in continental Europe, in France and in Switzerland, that it started to get recognized as a foodborne organism. And it has this particular trait, that this is some a newspaper article from the Franco-Swiss cheese wars related to Listeria. That when food manufacturers do get associated with Listeria, it can actually result into the death of their business, or potentially the death of their industry. Listeria itself, ironically, is a very low incidence organism. In the UK, there's only about 150 to 200 cases of invasive listeriosis a year, but usually about 20% of those cases do result in a mortality. So it's a low incidence, but a very high mortality organism. And it's got the unfortunate scenario that those deaths occur for those that are very young. So it occurs during pregnancy, and it can result in stillborn deaths, or in the very old. Generally, those that are in the 20 to 60 range, it's something that we can easily manage. In the UK, one of the more recent and larger outbreaks that actually put this on our strategic map at Quadram was the 2019 sandwich outbreaks that were in hospital settings, generally in about the Manchester area. There was 9 cases and 6 deaths, so a very high mortality. Again, if you're serving contaminated sandwiches to a highly vulnerable population, that's the result that you're going to get. But this is indicative of the actual issues of Listeria, in that it is more so related to post-process contamination. It found its way into sandwiches because it was in an ingredient in the sandwich. So whether it was the lettuce or the cheese or the meat, it's something that spills over during the production of the sandwich, or the production of one of those ingredients. It spills from the environment, so from the food production environment, into the food. Listeria is a little more rarely associated with a classical zoonosis, so coming directly from an animal product. It's more so related to the food production environments. This is, I've got 3 slides here, just stealing from Sanjay Gumela, who gave a great talk. He's the director of the American Frozen Food Institute, and he was talking exactly about that, the different food types. So chopped spinach, cauliflower, hot dog buns, pumpkin seeds. These are now the food types that are associated with listeriosis. It's not just strictly deli meats and cheeses, although those are still very, very, very high risk. And Sanjay went through, for all the businesses, where's your best money spent in terms of managing a listeria risk in your setting? And it certainly was, yes, have a little bit more sampling, do test at a certain frequency, understand your critical control points. Are they working? But then you've also got a lot of decisions to make in terms of a risk-based process. And this is where Listeria is going. So it's not just if Listeria is present, it's how much Listeria is present. There's actually a high load of Listeria that actually presents as a human health risk. For a lot of strains of Listeria, if they're at a very low level, the risk is going to be very low. That's what's been happening with these sandwiches, is that there's a very profound amount of contamination that's finding its way into the food, and that's what's leading to the health risk. So I'm going to go through a case study where we had the very fortunate scenario to work with UKHSA and a food business, which I'm just going to call Company X. Company X has had a very persisting strain of Listeria in their facility since at least 2017. So persisting that they called in UKHSA, which is a rare move for a food business, to call in the regulator to help with the sampling. So the genomics on the screen is actually coming from UKHSA, where they did multi-year sampling within the facility. Anything in green is the environment. Anything in blue is the food isolates, and red is a clinical case. So even though they've had multiple years of contamination in the environment, multiple events, hundreds of events where it spills from the environment into the food, there's only one clinical case. And so this shows again that kind of this different risk profile that's associated with Listeria. Not all of them can result in immediate illness. In this particular case, again, it was a high infectious dose in the sandwiches. 12 sandwiches, not in one setting, but during their inpatient setting. So they had a high exposure. Nonetheless, this is one strain of Listeria that's been in the facility since 2017. And for us at Quadram, this was the perfect example to work with a business, to engage with them on genomics, and to help them think differently about the problem. So they're spending a lot of money on their testing, Listeria. They keep getting their Listeria result back. They've brought in UKHSA, Listeria. So let's actually dive a little deeper into this scenario. Let's think differently about it to see what we can do to address the issue. And then once we have that, we want to act on those findings. Producers have already been kind of warmed up by their trade associations. Again, I'm showing the Chilled Food Association. This is a piece from their own guidance, talking to food businesses that if you start to sequence your Listeria, you're going to see more than just Listeria monocytogenes. You're going to see all sorts of other things. And it is great that their own trade association is providing some guidance on what will happen with genomics. In terms of Quadram, we've actually built this into some of our strategic lines that for enduring threats, this is what our role is whether it's Shiga toxin producing E. coli, whether it's Listeria. If it's enduring in the system, we can apply some technologies, genomics, metagenomics, metabolomics, and help people think differently. If it's an emerging threat, we can help government act swiftly, so we can fill some bandwidth gaps that they have. In terms of Listeria, just quickly on the genetics, there's two genetic lineages of Listeria. Lineage 1 is in kind of the turquoise, lineage 2 is in kind of the orangey color. And in general, those the zoonotic strains of Listeria, those that come from cows and come from ruminants, are in green. And being up in the top left-hand quadrant shows that they're more associated with clinical cases than they are the environment. So these are your so-called hypervirulent clones of Listeria, whereas in the orange, the ones that are more associated with food and less associated with cases are your hypovirulent ones. And the strain of Listeria that Company X has is the one that's right on the very bottom here. So clonal complex 121. And that explains why it's only one clinical case. So very, very successful within environmental niches, relatively unsuccessful when introduced to a mammalian system in terms of invasive disease. This rule doesn't always pan out because you can see that there's not a breakpoint between them. And the strain of Listeria that caused a large hospital-associated outbreak in the UK in 2019 was a CC8, which is it's an orange one. It's a lineage 2. It's right in the middle. There's also, as a Canadian, we also similarly had a large national outbreak of hospital-associated sandwiches in 2008, over 20 deaths, also a CC8 strain. So this isn't a rule that I would go by as a food business that if it's an orange one, it's fine. It's not fine if it's an orange one. It's really about how much Listeria is present in the product. So we've been working with food businesses to start to understand the microbial populations that they have. So starting to introduce technologies like whole genome sequencing. So helping the people identify within their system What organisms are actually there? Looking at the genetics of individual organisms, what do they contribute to the community that's present in your process? Using metagenomics to identify that entire organism. So who is there and how are they acting together? Honestly, this slide is not from a food safety perspective, it's from a food spoilage perspective. I'm working with a lot of businesses who, yes, they're concerned about the Listerias and Salmonellas, but they're more concerned about the Pseudomonas and the other organisms that spoil their product, that cause waste either in as they're producing it in retail settings, which they really hate. Obviously, if things go green or if they explode in a retail setting, that's very bad. Or if the same happens in consumer settings, more businesses are worried about microbial spoilage than they are about microbial food safety pieces. And this is where we can apply metagenomics with them to understand where those, again, spoilage organisms are entering into the equation. We've been also adding in a bit of chemical composition analysis. So it's not just the microbes, but what are they actually producing that relates to the spoilage, so we can understand how that community is working. So a multitude of projects that we have in terms of spoilage that have a metagenomic piece. I do want to talk about what we can do from a food safety perspective. And one of the businesses that we worked with in terms of us selling them on the concept of metagenomics, this is what they retorted with. And I basically have this up on the wall. I remind myself of this every morning. And from a business perspective, the last thing they need is an expensive list of microbes that they don't know what to do with. So we're not going to pat ourselves on the back because we've been able to reveal who is there in terms of the composition, but we want to help them understand how to actually address the risk that they have. So in talking with businesses, this is what we've started to curate as a list of questions. We're adding a little bit of our own scientific language in, but this is basically in their own words that they want to use genomics or metagenomics to understand, is there a predominant strain that's in their facility? Is it spilling over from the facility into the products themselves? Are there other microbes that are supporting that pathogen that's present in the facility? How is this finding its way into the products? How are these pathogens surviving all the cleaning and disinfection measures that we're throwing at this? We spend so much money on their hygiene and their C&D, and these organisms continue to be there. Can they actually track the spread of this microbe across their facility? Because you can imagine between the loading dock to the harvesting area, to the wrapping area, where is it spreading within the facility? Some are asking other questions like, are there high risk and low risk strains? Potentially yes. But also, what is just the baseline composition of the microbial composition that we have across all of this different piece? And also other pieces, if we are putting new interventions in place, chemical interventions, physical interventions, engineering interventions, this is actually having any benefit. Is there a shift in the after compared to the before? These are all answerable questions now. And so I'm going to quickly show the work of a couple of the postdocs on my team. So first from Anna Victoria, asking the questions, what are the genetic traits that drive the survival, emergence, spillover, and spread of Listeria in agri-food niches? And this is again using the case study of Company X, so that clonal complex 121 strain. Anna Victoria's drawn up a dated tree that shows this is basically the global collection of CC121. Groups like Institut Pasteur have a fantastic database that we can draw upon. UKHSA has their own short read archive that we can draw upon, and of course all the strains from Company X, which we see at the top. We can start to do a lot of munching and crunching on these genomes to try and parse out and find the genetic traits that are specific to Company X, if any exist. And the answer actually is a resounding yes. So these bars here show the different genes that map to individual components of the phylogenetic tree. So we can start to piece together what are the genes that are specific to different epidemiologic events that are happening. I'll skip through this except for to say that for a lot of these different traits, what has been acquired by the Listeria is prophage. So this is one of the major drivers of adaptation, of evolution in Listeria. It's the macro evolution of a large contiguous block of genes coming from a bacteriophage that's landing in the genome as a prophage. So here we've got a different way to view that gene content that we saw. I just want to highlight here this large blue block on the top right that's associated with the Company X clade. So let's have a closer look at that. And again, what do we find? We find a bacteriophage. So this strain that's been present for probably more than a decade in this facility. Its defining trait is a singular prophage that it refuses to be without. So this is the genetic trait that is driving success in that particular niche. So now we know why it's successful, but let's understand a little bit about the how. So this is something the team is probing right now. But within this particular prophage, there's genes that are related to biofilm formation. So there's pulling genes and a few other genes that are associated with biofilm formation. So that's what we're going back into the lab just to prove that these specific traits are quite valuable. Of course, not much of this matters to the company, except for saying that it does show, it gives it a little bit of the why. In terms of how this particular organism is surviving some of their cleaning and disinfection procedures, we're able to find genes for quaternium ammonium compound resistance. So that is providing resistance to some of the sanitizers that they're using. So let's have a closer look at the actual populations that are present in Company X. So the individual strain is interesting, but we'll probably get more answers if we look at the entire population. So Maria Diaz, one of the other postdocs on the team, is asking the questions: what are the natural populations of microbes that exist in foods or food production environments that support the survival and transmission of Listeria in these settings? Again, we're going to draw upon this really rare dynamic that we have to work with a food business and UKHSA. And we're able to go back into the environment and over multiple weeks, take additional samples from the facility. Of course, at this point, the environmental health officers at UKHSA know exactly where to find the Listeria because they've been doing it for years. So within two high care zones of the facility, high care is, if you aren't aware, it's food business terminology for where they're producing the ready to eat components. So they have to be very hygienic areas. In this particular facility, there was a prep area that was at 4 degrees and a production area that's at 10 degrees. So this slight temperature differential will become important in a second. Anyways, multiple samples across both sides of the wall here, predominantly in the floor settings, because that's where they knew that the Listeria was living. We did metagenomics, some standard cultures, and a few other technologies. And this is just showing the culture results, where in red is Listeria monocytogenes and in orange is other strains of Listeria. And so when we did this work a couple of years ago, sampling over multiple months, again, the organism was still present. So it hadn't been eradicated from the facility. So then we went to look at the actual metagenomes of those samples, realizing can't read this, but the whole point here is Listeria is not on the list. We could find a multitude of other organisms across all these different sites. Listeria not on the list. What it takes to find Listeria in the environment is two successive rounds of culture. Very specific culture enrichments of Listeria are needed to actually pull it out of the environment. In its natural state in the environment, as we see here with metagenomics, because this will be unenriched samples, this will be swabs directly from the facility with no processing, no microbiology. You can easily find a multitude of environmental organisms like Pseudomonas and Psychrobacter and Sphingomonas and things like that, but you can't find Listeria. But these other organisms are interesting too, because they show what is the flora of this environment. And what is striking is that it's very consistent over time. You'd think if you saw the facility, if you saw them clean the facility, so throw down like buckets of chlorine on the floor, scrub it, basically have the Zamboni machine drive over top of it, you'd think nothing can survive that chemical Armageddon that was just thrown down there. Nothing is going to survive, but those Pseudomonas survive, and that actually makes sense. You think of the microbial variability that exists in the world. Some are going to be selected for to survive. This isn't going to be a sterile environment, that over time you're going to have organisms enter and stay that are able to survive against that chemical plan that the facility is throwing at it. And that's what those Pseudomonas are. Now what's happened is the Listeria has snuck into that community and has become an integral part of it. So there's probably some sort of mutualism between those communities. This is just showing that between the prep and the production area, so the 4-degree and the 10-degree area, there is a slight shift. There's a slight difference in the population. They're related, but they're slightly different, and that would make sense that from a microbial ecology perspective, between 4 and 10 degrees, you're gonna have slightly different organisms that are gonna be successful. But nonetheless, very consistent over time. Even when we sampled, they did their deep cleans typically on a Wednesday. Take samples on a Wednesday morning before the deep clean, let them do the deep clean, take samples again, doesn't matter. Same flora are there on the same day. But again, they've been selected for. They know how to survive that chemical plant, but they'll be slightly different between the two areas. When you redraw these maps based upon the presence of Listeria, not where they're found, but just was Listeria associated with the sample? There's no association. That actually makes sense. If when the Listeria that is there, it's at a very, very minute amount. It's the Pseudomonas that's singing loud and proud about its presence. The Listeria is a very subtle member of that community, but it's not a driver of that community. But the other point that we have here from the sampling was, even though it's a minute member of that community, it still contributing something to it, because over the sampling period, you could always find that CC121 strain of Listeria, and on occasion, you could find another strain of Listeria monocytogenes, including sometimes some of those hypervirulent strains, the ones you'd be really worried about. But by the time you came back the next week and sampled again, those are all gone. So they get evicted from the facility. So we've got our persisting strain, which is contributing something to that environment, versus the evictors, which obviously contributed nothing, hence why they're gone. So what is that persisting strain contributing? And that's where we go back to the other piece of data that shows it's probably contributing to the overall fitness of the biofilm that's been established. So that's part of the dynamic that's happening here. Okay, so let's end on some of these questions. So yes, is there a predominant pathogen in the facility? Yes. Are there other microbes that are supporting the pathogen? Yes. Do we know how the pathogens are surviving the disinfection measures? We do, because we know the genetics of it, and we also know some of the biofilm pieces. Can we track it? Yes, we can. Can we put some risk piece on it? We can do that as well. Do we have that baseline composition? The answer is yes. It's easy. So it was great to answer yes to all of these things, but there's no a yes isn't action. So can we help the producers go from what they formerly had, which is really expensive microbiology test that just says it's plus or minus, and now okay, we've come with our beloved genomic solution, but does it actually provide any benefit? So we're working with food producers to try and get a perspective of what would they actually change based upon this information. And so the first thing that they can change is their chemical disinfection plan. If you find out that there's resistance to what they're using, they can shift to another, or they can shift to a higher amount. If 200 parts per million chlorine isn't working, 400 might. So these are things that they can adjust. But they can also look at what we're calling facility-specific pathways. That again, there's multiple areas of the facility. You might get to know how that organism is moving it around. Is it coming on equipment? Is it coming on shoes? Is it coming on a particular ingredient that's going through the facility? If the answer is yes, then you can break that chain of transmission by understanding that pathway. You can also use innovative materials. Again, this is where the engineering controls would come in, that these microbes need something to stick to. You figured out the pathway. So now, yes, you may be able to break the pathway by changing the way equipment moves around, but you might also just need to change the equipment itself too, because Listeria can embed into particular components. Actually, my favorite slide from Isopol, so the international meeting that we had a couple months ago, was from Nofima in Scandinavia. So they're a research firm that helps. I think they've got a specialization in seafood production facilities, and they were working with a facility that had a persisting Listeria on the salmon skinning line. And at the end of the day, the belt that they used for the salmon skinning line was so embedded with Listeria, they had to discard the entire machine. There was no way to clean and disinfect it. And that's kind of a common thread for unfortunately, it's a common thread for a lot of food producers. So if you get Listeria into the facility, you're probably going to have to dispose of the equipment that's contaminated. If not, some people get so frustrated, they just say, we're going to have to just burn down the entire place and just start from scratch, because it can be so hard to get rid of these things. Nonetheless, on to the fourth, some people are talking about novel ways to shift the microbiota. We're talking about biocontrol here. Biocontrol is used a lot in agri-food. Can it be used in food production settings? Can you introduce bacteriophage or other competing organisms that would actually suppress these growth? Can we use things like non-chemical interventions? Again, we saw this at IGS. Profound use of UV lights as a routine control measure. So they don't have that opportunity in traditional farming settings to use UV light the way that they're doing there. So it's fantastic use over there. Fantastic use of hydrogen peroxide. I'll skip the rest. But just to say that this has all been a learning process for ourselves with these food businesses. And at this point, to be honest, it's still all very proof of concept. This is not something that we're in a place to routinely apply, but we're having more and more food businesses come and knock on our door, whether it's a spoilage question, whether it's a pathogen question. Food businesses are starting to recognize that, again, there's only so much information they can get from the traditional testing. There is a value, there is a place for some of the metagenomics. So again, I'll just thank my own team that put this together. I missed out some of the references. Maria's paper on the Company X metagenomics has been published. Anna Victoria's paper on the Company X comparative genomics was just submitted, so we hope to have that soon. But I can provide those to anyone that's interested. Certainly want to thank those at UKHSA, because these studies are only possible by the accessibility that they gave us to Company X. And just again, my team at the front there. So, but Raman, thank you very much for the opportunity here today.
So any questions? Anyone online or anyone?
Yeah, I mean, I was quite interested in this concept of using biocontrol in a food setting. Right? I mean, is that really, you know, are the authorities ever going to allow that? I mean, beyond phages. I mean, phages clearly, right? But if you're trying to look at competing microorganisms or something like that, for example.
So some businesses are absolutely using it on food.
Okay.
In the environments, I think that'll be a stretch, but on food.
Yeah, during production, right? Yeah, okay. But post production processing, that's not a reality or at the moment.
No.
Yeah, okay.
That's more of a think tank piece. That again, if you have, so you can even see the conundrum here for Company X.
Yeah.
That again, they've got, unfortunately, a very well-established interconnected community of microbes that are in their facility, and unfortunately, Listeria is part of it. That Listeria is also evicting out the really bad Listeria. So, in a way, one could argue they've got biocontrol against the worst Listeria.
Sure. And I mean, I guess a related question. So the reason you're getting that chemical resistance is due to biofilm formation, right?
So it's intrinsic in both the strains themselves. So Listeria can readily acquire tolerance to biocides. Here we see it genetically with the transposon related to that literally gives you resistance. You have a multitude of other mutations that just arise within membrane genes and some other components that also give you resistance. So and tolerance to biocides is certainly possible. But then in that community structure of biofilms, does that enhance the resistance? Potentially, yes.
So it's this treatment scenario, a hamster wheel, you're always going to be chasing. It kind of goes back to some people here that work on phytophthora. We chat about this. The bug will evolve and win every time.
Yeah.
It'll keep you occupied for the rest of your career, obviously. The interesting bit was I was thinking less about the science, but more about chatting to the companies about getting their data. How long did it take you to get them to play nice? Did they open up quite quickly?
In terms of the business-to-business data sharing, so that's we're literally having the next planning chat with 5 businesses that have committed both resource and time and obviously strategy to help develop it. But it's 5. I've talked to many, many, many more than 5 businesses to get there. And it's really there are businesses that again, they invest a lot routinely in their own facilities in terms of interventions, engineering controls, data acquisition, data management tools. I've been to facilities where, you know, the plant manager can tell you the average kilo of chicken carcass that's flowed through the abattoir line in the last 5 minutes because they're making the measurements. And these are the types of businesses that have that culture of food safety. We're less worried about the risk and more worried about the opportunity to share data that are signing up.
So spinning risk into confidence, which is, in a business perspective, is 2 sides of the same coin.
Yeah. One of the businesses that we spoke with, his line back to me was, why would I ever want to hear about the food safety issues that are happening with my competitors? Because then I got to go look for that too. Right? So that just gives you a window into a different type of mentality. Yes, there are businesses that want to spend as little as possible on their microbiology. They don't want to. There's a bit of a head in the sand mentality. But I'm not admonishing them because they've got a multitude of other pressures that they're trying to manage. It's going to be a select few businesses that have made that they're going to invest in the science to control this. You can see it in the facilities just in terms of the signage and the educational materials and all the other supports that they give their own staff to make sure that they're producing both safe and high quality foods. And usually see this in a business, just even their titles. You'll see quality and safety. You have a director of quality and safety. You know that they take it seriously. So,
are there any incentives to get businesses involved in your intelligence platform? Like, is it going to be subscription based for people to get the data, or if they upload data, then they've got access? You definitely have to pay to play. So right now we're just going through a pilot phase with the 5 businesses. Can we even build it? Can you even harmonize the data from 5 food businesses? Hopefully the answer is yes. And hopefully the answer is yes, we can learn something from it. But in terms of the value, so yes, ideally, this is a tool to understand and manage where risk is present in the system. But you can think of the economies of it in that, again, if there's 6 of us in the room, if we're not all going to test things the exact same way. We're not all going to test the exact same ingredients and supplies. So we learn there's a lot of value in learning what's happening with some of your competitors. And if let's say we all had this shift to a new supplier of sunflower oil because of whatever's happening in the world. That's a recent example. Everyone's got to shift because of geopolitical realities. Everyone's got to shift to a new supplier. Let's say you've tested it well, and you found it to be safe. That's actually good information for the rest of us. So you get kind of that scaling, that's of value. So you know where risks aren't in the system, but where they are. You also get a chance to understand where to de-escalate some of your testing. Because for a lot of businesses, they'll be investing testing into areas because at one point there was an issue in a particular ingredient with a particular pathogen. And so because of that historical incidence, you're still putting a lot of money at testing that. And maybe you're finding nothing but negatives. But again, you're not reached the spot where you're confident enough to de-escalate away and say, we're not really going to test that anymore because we don't think it's a risk. Again, if collectively the 6 of us in the room saying, yeah, we're all testing the same thing for the same reason because we all thought it was bad. But for the last 3 years, it's been perfectly fine. Well, then you can start to de-escalate. So it'd be the same principles as what happens in healthcare settings, in infection prevention and control. is that you learn where the hot spots are and where you need to put a little bit of energy, but you also learn where the cold spots are, and you can actually de-escalate away a little bit. And that gets a little bit more valuable in a healthcare setting when hospital to hospital is starting to share that information. Because of course, these organisms are not just unique to one hospital. They're going to be transmitted between them. So that's why hospitals already do this. This was actually our case study to show food businesses that you can actually share what is otherwise pretty confidential data without breaking a system. You actually can help each other. So.
I'm interested in the use of metagenomic for identification. I mean, following if they have been late or not. What is it? How do you fit the threshold of that? How do you determine it's one gene is enough, several genes, several bacteria at the same time? So you can identify, but when you take action.
So from the metagenomics, again, this is where I close with that remark. Be careful with myself that this is really a proof of concept. We've got one business where what they make is a live cured ham product, and taken a lot of work to figure out the actual reasons within this very complicated system where the spoilage actually amounts to give the candidate organisms and the candidate mechanism of why that particular product may spoil. It's worth 10 seconds to talk about it. So for live cured ham, they brine it in a brine that's probably been going for decades in a tank that's probably about half the size of this room. Open air, half the size of this room. The whole point of the live brining is to microbiologically introduce nitrates and flavor into the product rather than chemically introducing nitrates into the product. But they've got the brine that's been going probably for decades, and they've got no clue what is the microbial composition. There's going to be obviously a lot of anaerobes, a lot of fungi in there. But being a live brine product, even though it's cooked, there's still going to be some of those microbes that carry through to the end product. They can result in spoilage of the product because they're lying there. But yeah, it's a lot of work to go through. What is the composition of the brine? What is the composition of the cooked product? They deep shell it, they slice it, they pack it, and so the microbiology shifts at every step. So it's honestly been a lot of work with that company to pinpoint down to the specific genes, specific organisms that might be involved in that process. That's where we had to pull in other disciplines. That's where we pulled in, worked with RSSL to understand the chemical composition. What is the actual metabolite that the organisms are producing that's resulting in that shift? Because the genomes itself wouldn't tell you. So.
I was in the case of a vertical farm or greenhouses, it's more easy to determine. It's like a harvesting point before passing to the problem to someone else. That will be the time to analyze whatever is happening there.
But you don't know if you've not looked.
Yeah.
No, that's why if you want to look, that's the time. It's more easy in a vertical farm to analyze. If you want to analyze something that a food processing that there is multiple steps, and like he said, it's changing along the time.
Yeah, like a vertical farm would be a good example because again, there's a lot of, you just saw it, there's a lot of really well thought out processes that like from the germination all the way through to the harvest with all the irrigation steps and the lighting and everything that happens in between. And yeah, it's conceivable that again you've got this environment that's by definition it's under control, and IGS is to give them credit. Not only do they have it under control, but they actually actively manage that control. Like they think actively about the water supply and the air and the substrate to make sure that there's no chance for a biological problem to kind of manifest. But it's possible that something could. If something was resistant to hydrogen peroxide and resistant to UV, there's certain crevices in there where it could creep in. But again, that's where maybe the metagenomic sampling would help you start to understand where within that line is that organism being introduced and surviving. And then you could add on the more specific genetics of, well, how is it surviving? all of these great control measures that they otherwise have in place. So
I think in food production as well in the industry, they're not always producing the same thing every day. So it could be modular shifts in of the kit. Most of them will build them now, so one bit can come out and another bit goes in. So if you treated that production line, that's fine, you've controlled it. But if you're swapping it out for, actually, we're going to make biscuits instead of sandwiches, it's an extreme example. Different feedstocks are coming in with different problems, and a different, you'll take one module out, put one in. So you've changed it, your control mechanisms will be different. Now, vertical farming can be that by growing something different. The other thing is that one of the drivers for change could be you talk to the big manufacturers who have got multiple different feedstocks coming in. And the oil example was good. Probably the other example would be when the price of butter went nuts. So suddenly you were going from UK butter to Dutch butter to Scandinavian butter. They won't all be the same. And so your existing process you thought was great might not be fit for purpose now. But the large processors exercise control because they demand it of their ingredient suppliers. So it's they're rather than policy, they're exercising control with this.
and that's a huge strength to the UK system, is that the major retailers and the major brands do exercise that buying power and do exercise that. If you go to some North American examples, you think a company like Walmart could do that. And I think for certain components of their business, they do exercise that buying power. But I think on the food safety side, that they do struggle. And this is where one of the projects that we sorted out within the Food Safety Research Network, some of those UK companies that do have that strong buying power were getting challenging answers on a certain food commodity, and that was low moisture ingredients.
Yeah.
So seeds and nuts and spices. So this was a project that was less about the microbiology, it's more about the social science piece of it, because people presume low moisture ingredients are microbiologically safe. So the producers of those, I'm not casting a wide net here, but some of the producers would challenge a retailer. Well, why are you asking for my certificated testing on this? Because it's red pepper spice or something.
Yeah,
they're asking because there is a large risk there.
Mm-hmm.
So yes, it's good that the retailers and the big producers here do exercise that buying power, but it's not universal across all product classes. So
good to hear. So unless there's any other questions, I think we'll draw that to a close. So thanks, Matt. I'm sure you'll be in touch. There's interesting collaboration points there.
yeah.